Ma'am, we can start. It's 6 p.m.
Okay. Okay. So good evening, everyone. Today, as yesterday, we have discussed about the diversity analysis. So today I will again start with that left part. So we'll be starting, you can see my screen. We will be starting with the structure analysis.
Yes, ma'am, your
So,
screen is visible.
yeah, so all of you have downloaded structure. So the first step is to open the new project, then stepwise you can see what it is doing. I will explain you again. Just to see this video once, then I will explain you. हेलो हाँ जी अच्छा हम्म ओके ठीक yes we can start with this. You can see my screen, you can hear my voice.
Yes, ma'am.
Yes, so in this first slide is explained that you will open the project, you will take the project name, select the directory, and then you will uh, write the name of the project and where it is present. You will browse the file which I have given you. Okay, everyone do this step, then we will go to the next step. And then you will input that 329 individuals are there and the priority level is 1 for sorry, in your case it is 10 individual, 10 priority level, and 10 will, and -1 will be the marker. I will show you in here also. It will be more comfortable to use. See, I have run this project already. Open data, you will open the data wherever it is, wherever you have saved it. I have saved in my C folder, GWAS training structure. The name of file is structure input. You will open it, then it will tell you that how many columns and line it has. Because I have already uploaded it, so it has open. You have to enter 10 markers, 10 loci and 10 individual, and then you have to say missing marker is written mentioned by -1, just like I have shown in this in zip file. So this way you open it first, then I will explain you the next step. Everyone, I feel, must have opened the file and entered that there are 10 samples and 10 marker, means 10 individual and 10 marker, and missing genotype is, so here in number of individual will be 10, polyploidy 1, and number of loci will be 10, and missing value will be -9. Then you go to next and then select row of markers and then number of individuals for each individual. Then this file will come and you will press proceed in it. Okay, then you will get this data. Then you will get this file open. The next step is that you will go to the next step will be that you will go to new parameter. In that, burn value you will be giving is 3, and the burn that is length of burn period is 3, and the number of time replication is 1000. Once you give this, it will ask a name and it will start run. Okay, this thing you can see repeatedly. It is showing that once you have uploaded your structure file, will give the burn length and the replication. It will start the process of how much you have given. Okay, how many burns and how many replications you have given. Once this is done, the next step which happens is that we have to start the job. To starting this job, it will ask you how many k value you want. So you write 2 to 8 or 2 to 5, because more you will give k value, it will take more time. And iterations also you can reduce, because here we have mentioned 10 iteration, it will take more time. So to run faster on your system, you can give it more iteration. But for a ideal result, it should have at least 1 to k value should be 2 between 2 to 8, and iterations you have to give at least 10. So for ideal, because we are just doing all test data sets, so this is not the actual run which we are performing for a study, but for actual you have to do this way. Once you do give the structure scheduler this option, it will start running. Okay, after running the results which we will be getting, now I will be explaining you about those results. So see, you will get these results which are shown in the left hand result, these file. Once you run, you go to the plot, it will plot the structure of your, means how many populations are there. It will plot, like we have selected K2, it shows 2 population. We are selecting higher K, it shows more number of population because it is showing 3 colors. So is this part clear to you? Then I can go to the analysis, or I should repeat it. I will repeat it on my structure. See, I have also run this. Like I'm seeing I have run it with K1 to 12. It has given me all these result. When I check the plot, how is the plot at K2? If you see the bar plot, how the structure division it has given that there is at K2 there are 2 subpopulation which it's predicted, and how is it value? How is it there? What is the trend there? If you see at 3, it will show the 3 subpopulation within the population total you have given. If you give it 5, it is divide your, it will show how you did, how is the trend when you give it 5 subpopulation. This way, now you have got your results at each population. So you can this is the FST value if you want, and alpha value it will also give if you want for your data.
Ma'am, your
Because
screen is on association analysis. Is it at right place?
no, it is not at the right place. Now I have opened the structure result. You can see it.
Yes, ma'am.
Yeah. So if you see, if you want to check that this is the result which I got after running. So if you want to see what was the result at K2, like K1 we will not take because K1 is the state when there is no subpopulation. At least one subpopulation we will consider to have. So the result you will see that it has shown that your original population is divided into 2, and you can also get the FST and alpha values with here. Then if you see the result at K4, your subpopulation, here it will show how your data is divided into 2 subpopulation. Now when you get this result, now to decide which K value is best, you will take this output to structure harvester. It will give you what is the peak point where you get the best K. So all these results will be imported into structure harvester, and it will show you which is the best K. Okay. And here it has given the information that what we have used is that during our run, we have used 10 individual, 10 loci, 4 populations, and 3 burn-in period, and 100 replications. Okay. And then it has told that overall proportion of the membership of the sample in each of the 4 clusters is there are inferred 4 clusters in which 1, 2, 3, 4, and what is the proportion of the membership of sample in 4 clusters? How much? If you say 4 cluster, then 0.23 fall in one cluster, 0.26 fall in other, 0.285 fall in the third cluster, and 0.224 fall in the fourth cluster. Then it tells you there is more information about the overall allele frequency. How is the allele frequency divergent among the population? Because we have used very less smaller set of data so that our output is faster. Therefore, it is showing very low allele frequency, and the expected heterozygosity is also not so high because we have taken a small data set. The actual data set will be of thousands individuals, more than 100 individuals, and many more markers. Lakhs of marker could be there. Why ploidy is set to 1? Because I have used input data which I have given you. It is, I have taken the haploid data. It is not the diploid data. That's why I have set ploidy 1. FST is a measure of diversity, and I will tell about Q matrix when I will come to association mapping. Okay. So the next part is that it will give the estimated probability of data. It will give you the mean likelihood, the mean FST values at for each population when it at because this data we are seeing at K4, so all the results are at K4. How much is the percent of missing data in each subpopulation, that also it will give you the the missing information it is giving you at which locus the missing information is for which marker which we have in our input data, it is also giving that information. So finally we have completed this structure analysis. I feel all of you have run at your end. Then I will be moving to the next part. Yeah, genetic differentiation, which is basically representing the diversity among the subpopulation. Genetic differentiation only creates the diversity. Okay, so I'm going to start association analysis. I feel all of you must have downloaded Tassel software. So burn-in period is a parameter which is used to tell that how many times you want to run your job. Means burn time, how, what is the length of the when you're giving multiple replications, what is the length in which this k value iterations are to be made, how much is the burn length. So now we will go to Tassel. We will read little bit about, first I will tell you about Tassel. Tassel is a very popular tool in this in GWAS studies. Tassel has been developed by the lab. I'll just open the Tassel. It is developed by Berkeley's lab, which are very popular in working on GWAS. They work on maze. They have also developed a software called Gapit, and this Tassel you can, which I feel you must have downloaded according to your whatever is your machine. Okay, and how this Tassel helps us to, there are many of the Tassel also has option of filtering data, alignment view. It runs all the models of GWAS can be run on Tassel.
Ma'am, sorry to interrupt you. Your screen is not visible.
Okay.
I mean, slide is not.
Okay. So Tassel is helpful in making the QQ plot, Manhattan plot, and this is the Manhattan plot. If you want to make QQ plot, that can also be made using Tassel. And Tassel is very user friendly because it is just click based at many steps. There is a command line option also of it, but that is just for Linux, and you can do phylogenetic, genetic heat maps also can be made using Tassel. So first I will start the association analysis and then explain you. So I will go to Tassel. See, I have done a bit of the analysis. I will explain you how I have done. First, you have to go to file. I will reshare my PPT. Yeah, you can see association analysis. The first step you have to do is that you have to import your genotype and phenotype data. Open the genotype and phenotype data, go to the folder and open the genotype and phenotype data which I have mailed you for the Tassel analysis. Once the genotype and phenotype data is open, you can individually look at the data. It gives the information that what is present in your phenotype file. You can visualize it. What is present in the genotype file, that is also exactly visible. And then comes the see, you can see the taxa and pH values there, and the next come is that now what we will be doing, that we will see that what is the trend of our our phenotype, how it is the pH distribution of our phenotype data is it is a normal distributed data that we can by using the select phenotype and go to analysis and then run the histogram option. You will get this pH distribution. Okay. This will give you the histogram, your trend of phenotype, to know how your is trend of your phenotype. Someone is asking something. We are not using GAPIT for GWAS. No, we are not using GAPIT here. We are using Tassel. So then comes the next step is that now if you want to, we have looked into the phenotype data. Now we want to look into the genotype data. So we will be going, how we'll be looking to genotype data. We'll select genotype, go to data, create genotype summary. It will create the genotype summary. You will be able to see all the genotype overall, how many individuals are there, what is the allele summary, what are the site summary. You can get all of that, and you can also plot the histogram of the allele frequency. Your site summary, if you want to plot whatever you want to plot, you can plot using the chart option in the results. Okay, it gives the option for all, like this is chart for the minor allele frequency. If you want to plot some other aspect, you can plot everything of for your genotype data also. Similarly, what we plot for the phenotype data. So we will first select the hello.
Phenotype is having a skewed distribution. Can we still perform, or you have to go for a transformation?
No, we can perform because genotype is basically we have to identify. The aim is to identify those allele which are in LD. So genotype distribution will not be a uniform.
I'm asking phenotype. Phenotype, if it is not normally distributed, sometimes skewed distribution will be there. No?
Yeah, that can be. Ideal data is a normal. If it is not, then you can go, but the number should be high. The number of individual which have been phenotyped should be high. Phenotypic variance should be good.
Yeah.
Variance of the phenotype should be good. This is important. Okay, you understood?
Yes, ma'am.
Okay, so this step I feel you all have understood, that you have to make, how can you make the different plots to understand how is your data set. So now once we have understood our data set, the next step is we can filter the data set based on different parameters, which I can show you here. If it is the need, here it was not required, so I have directly went to an association study, but the how you can filter, I will tell you that.
Hello.
Hello. Yeah, please say.
I have a question.
Yes.
I would like to know. Yes, the Tassel. Which method is using to analyze the variance components for individuals to come up with an association?
We are using GLM model today. GLM model.
GLM.
Here
Okay.
you will see the model is GLM.
Okay.
It takes the matrix.
Okay.
Okay. Yeah. Say.
Ma'am, GLM, MLM, in which situation we can use them?
See, GLM model, what happens that when we run our analysis, GWAS analysis, if we have kingship matrix, if we have, then we run, it is a covariate of relatedness. Then we use MLM. If we do not have if we do not have kingship matrix and with relatedness value, then we will go for MLM. Sorry, GLM. Because GLM uses this, it will take the sample matrix and the phenotype and genotype data, but MLM will take genotype, phenotype, and the kingship matrix. It will need this. So see, actually if you for this to be very specific for the model, you have to read about each model. Every model has its own advantage and disadvantage. So
yes.
It is complete. Hello.
Kinship matrix, where do we get the values? Which tool?
I will explain you. how to get kingship matrix. See, once you upload your data, as I have told you that you have uploaded this sample phenotype and the genotype. Once it gets uploaded, now if you want to make the kingship matrix, just select the genotype and then go to analysis, and then here you will get kingship.
Okay.
Okay, and if this is for MLM, if we want to run this GLM which we are running today, in that we have to make the data distance matrix. So we will first make the distance matrix and then run the MD, that is the multi-dimensional scaling we will be doing. Some people also do PCA for GLM can also be done. Like if you do not do this MDS, multi-dimensional scaling, then you can also go for PCA. Because 3 files are required for running GLM is the genotype, phenotype file, and the either multi-dimensional scaling or PCA plot, whatever you get better.
Hello, ma'am.
So, yes.
Ma'am, do you have any paper so that we can get out of confusion from GLM, MLM, and CMLM? Because several times.
Yeah, there are many paper. If you
have-
search, I will mail you. I will send you on your mails. So there are paper which are only myself. I have a review in which there is a comparison of different models. Like there are many model GLM and MLM and farm CPU. All these model are getting developed day by day. You can see that what is the advantage of one model over the other.
Yes, ma'am. Please
Okay.
share if you
It
have,
is-
because several times we got confused in this.
Yeah, and sometimes I have seen when people are exactly they're very new to this field, then what they do that they initially run all the important models which are highly cited, and then they conclude that which set is actually giving the right trend which they expect. Okay.
Yes, ma'am. Yes, ma'am.
So
Please.
now as I told you, once we have genotype phenotype file, we have checked the trend of our how is the phenotype data, and also we have checked the trend of how is our genotype data. We can now filter that data. If you want to filter it based on thin site by position, this you can use this option. Give the output file, what the minimum distance, like which how much is the distance. Like yesterday I told you, between 500 base pairs or 1000 base pair or 50 base pairs, that region according to your data set. Then if you want to do filtering based on, there is a set low depth genes to missing, that is those genes which are having missing genotype more marker, those can be removed based on, like if it have 50% missing rate or 10%, 20%. It is all based on how is your data. If your data is very high missing rate, then you have to increase the cutoff. If your data is low missing rate, then you can use a stringent cutoff. So I think you got this point. Hello.
Yes, ma'am.
Yes, and then the filtering can also be performed, and if there is a lot of missing gene markers, imputation is performed in that case. And you can also filter your genotype based on taxa sites. Here you have to give the minimum allele frequency, at which you want to filter. What is the maximum allele frequency, how much heterozygosity you expect. If you want to remove the minor alleles, or you want to remove the indels because you are just interested in SNPs, and in which specific region you want to include, all that can be done with this filtering. If you want to filter it by the taxa table taxa, then you can, here it is mentioned that minimum proportion of site present should be present in that taxa, means that individual, how much is the heterozygosity proportion and how much is the heterozygous proportion minimum and the maximum. That this is actually 100%. This is we are not expecting any heterozygous minimum is at least it should there is least value, and means here actually it is 0, but we take it. Here we take 90%. Generally we take 10%, and this we take 100% heterozygous can be included. Then the other thing filtering which we do is trait based filtering, but site names can also be filtered, all these filtering is also done according to your data set. Then here is also an option of diversity analysis, but I have not used this option because I have used structure, and structure is more popular with it. This will also give you the FST values and all if you want to do in Tassel. As I told you, if you want to plot the relatedness distance matrix, create trees, merge trees, and if you want to make subtrees, all can be plotted. PCA can be done from here. Now the next step comes is association. So how to go for association, that I will explain you. So for the association, what you will be doing is you will be first selecting the genotype and the phenotype, just one step back. You will go to select genotype, first make the distance matrix. Once the distance matrix is plotted, you will go to analysis and then go to MDS plot, multidimensional scaling, and then it will create you two files for you. Those will be, as you can see, MDS PC matrix and MDS eigenvalue matrix. So this way you will create the multidimensional scaling file of the distance matrix, and you already have the genotype and phenotype data. So this is required for running because we are going to run GLM. So this file you have to create. So once you have created this file, the next step comes that now we have to intersect those files. So we will be intersecting the genotype, the phenotype, and the MDS file, which we have created for MDS PC. PCS file, which genotype file, which we have created just now. So once this file is combined, the intersection you have to keep in mind that you have to intersect. Without intersecting, you do not have to make union join. And if your data set, like here you see that these are the number of taxa 1030 and 41082. So if you have 1030 genotype and you do not have, you have lesser phenotype, it will not work. It will say that there will be unequal number of individuals to make for the intersection. So once the intersection is done, as after the MDS PC, you are seeing that it gives the different PC1, PC2, PC3 values. And now once the intersection is done, our next step is to run the association. So as we have seen that we how many sites are there, which are all the information also we are getting downside. So the next step is we will go to analysis, go to association. The next step will be we will go to analysis, go to association and run GLM. It will ask you that where is your input file. It is already selected. It will run the GLM and it will add the this information. Output will come, in which I will show you in my data also. It will tell you the and then you can plot the QQ plot by selecting the output. You will plot the QQ plot. The QQ plot basically is the log 10 value versus the position, and you can also plot the Manhattan plot. These are two most popular plot in GWAS analysis. So you can see here, this will I will now explain you in detail how is the QQ plot and Manhattan plot. What do they really give you, the what do they explain, what is the interpretation? First, all of you have you are able to run GLM. Hello. Yeah. So I will be now explaining you the output in my results. See, when I have run, I also got the similar result, which I've shown you in my PPT. This was the after running the association GLM, it gives us the MDS PC's matrix of the input data which we have used. It will tell you this was we have currently, because there were many traits in our data. So it has given the values for all the trait. The trait, what was the marker? What was the marker position? what was the marker probability? Hello? There is someone who is unmute. Please mute yourself. Then it will give you the marker R square value. And this way, this the probability, the error value, the model MS minor observations, all values it will give. Now because we cannot understand, we cannot go in depth of every value. So the plots which are helpful to understand this data is the QQ plot. When we have to plot QQ plot, it will ask which trait you want to plot QQ plot. So you have to select that this plot you want to plot QQ plot, and then you say okay. So it will plot this, give you this plot in which the X and Y axis, the X axis give you the expected p value, and here these are the different traits which have been mentioned here. So this black line is that what was the expected p value of the trait of a particular trait. This is the ideal state, and these different line represent like ear height. This is represented by red color. SHL is another trait by blue color. ASI is another trait which are different color. So this shows basically that what was how is your probability of getting the trait associated of the loci which you are plotting with the trait compared to the expected trait. So those points which are out of the line are those significant p values which are expected that these variants, because those which are falling in the same line, they are the chunk of SNPs which have a similar trend of LD across the majority of your markers in majority of the individual. But these individuals which are exceptionally falling out are important to be observed. And the more clear, one of the most famous plot is the Manhattan plot, which is based on the name of a building in whose shape is Manhattan shape. So if you want to plot, like we will be plotting together for all the traits so that it will look better. If we plot separately, then it can give you separate also. Like here it has given it, this is more clear to and easy to understand. It has given you, yeah, it has given you the position at which the markers are present and what is the p value, log p value, which indicates low, or means the minus, because it is log 10 p value. More the minus log 10 p value, that is the higher this you see the locus, the position at which your marker is present, it is more minus negative p value, it is more significant. So that SNP is more of our interest. Those which are not, these are the common one which do not have such high p value. So these can be, if studied in depth, in their function can be studied. These can be be the expected variant or the SNP, because we are studying, which have significant p value and they might be associated with the trait statistic.
Chromosome wise Manhattan.
Hello?
Chromosome wise, madam. It was based on positions.
Yeah, it is. We are plotting the positions.
Based on chromosome positions.
Yeah.
It was not there actually. It was based on just a position. I wanted like all the chromosomes.
Okay, I will plot that also. I've shown you here how it is getting plotted. See how it is getting plotted. Once you have done this analysis, it is complete. It will show you how it is getting plotted.
Ma'am, is there any criteria to select the optimum p-value for SNP validation?
Optimum p-value?
Yes, that we can
corrected.
say it is significant.
So see, it will only be, it can only be decided once you see the QQ plot or the Manhattan plot. Without that, how can you decide?
How many?
Depends. See, what is your cutoff? Means cutoff will be only based on your output data, na? And always 0.005 p value is for 5%. If we take corrected p-value at 5%, then if it is lower than that, then it is more significant in a general statistical term.
Yes.
Like for a RNA-seq data or for any data set, we take at 5% is 0.005 and at 1% it is 0.01. And
Is
if
there
you have
any-
applied the Bonferroni and all these corrections, then it is more significant.
We should also consider R-square value.
Hanji?
We should
Yeah,
also consider R-square.
yes. Yeah, we should consider.
Ma'am, why you have not used population structure output Q matrix file here?
Structure file can be used if we do not use this matrix, because I have generated the matrix here only, na?
Okay.
You have seen that I have generated this matrix.
Okay, that can be.
Yeah.
Then we don't need structure also.
Yeah, that is true. But I have made it because some people take structure input only.
Yeah.
So everyone, please, if you have any query on Manhattan, please take, because Manhattan is a very important plot. And it should be understood by all of you. And you must have all have run this, that how to intersect and then get the intersected file and then run this analysis, this GLM analysis. I feel all of you have got. And you also have run the QQ plot and this Manhattan plot. I will again once show you so that you don't have any problem. Someone has a query. Explained. See, in Manhattan plot, there is position of the of your p-value is corresponding to the position of your variant, your SNP it is plotting. But Manhattan plot is plotting that thing, but QQ plot doesn't plot that. Manhattan is, if x-axis you see of Manhattan, I will plot and tell you. Sorry. So we have to always select the stats value, because in the stats, the p-value is present.
Ma'am, you have taken haploid data here, right?
Anji?
You have taken haploid set of data. Or,
Yeah, we have taken HapMap data.
Have heterozygote SNP, how do you consider, ma'am?
See, many studies I have seen that heterozygous in those plants or in those organisms where the heterozygosity level is very high, then some percent of heterozygous alleles we keep. But if your heterozygosity is not so high and it doesn't affect the number of markers, if you remove them, then and in many studies you will see that they remove the heterozygous marker.
Okay, otherwise we can take diploid data also, right?
Right.
According to which one is better, ma'am? Haploid or diploid data?
Anji?
Which one is better according to your experience?
Comparing what?
Like haploid data and diploid data. I have both haploid and diploid data.
Diploid is better always. I was telling about heterozygosity. I was not talking about haploid.
It's always confusing. If you have heterozygote data, it is difficult to take haploid data.
Right. So diploid is generally taken. Haploid is not taken because we consider both the alleles.
Yes.
So finally, diploid is only taken in majority cases. And if you have a polyploid, then that is a completely different case.
Can we do that? Polyploid here?
No, majority of the algorithms of calling variant also, what I have told you, were based on only diploid data set. But there is an algorithm I feel you might be knowing, that like its name is FreeBase. FreeBase can identify a polymorphic site also, means, sorry, not polymorphic, polyploid genome also, polyploid based, not polyploid based variant calling also. But it is really complex, and if you are a starter, then you should not start with polyploid. Okay, someone has asked me. Kindly explain the criteria for p-value selection. How to represent the cutoff line on Manhattan plot for significant SNP. Okay. Actually, that the cutoff line which you are saying is no, that can be easily plotted when you are plotting this data with R, because I have made an R that by importing this data. But here, because it is just a GUI based tool, it will plot according to what is there, means normal trend. It is not plotting that way, that it will plot it making a line. But if you see for that, if you go and see for QQ plot, it has by default prepared it. We don't have to say it. If you select all the traits, see, if you are selecting all the traits, it plots all the traits, and it also gives the expected, this is the expected, the cutoff value above which all of our traits are coming, because this is a very good data set which have been provided by the Tassel software. So, but in that, for that cut, for making that line, expected line, which you cut off line, which you told, that you need to go to RBase tool. So someone has asked me, please, criteria for p-value. I explained already p-value. Please show how to represent the cutoff line in the Manhattan for that I told you. And then in our QQ plot, the curves for this some traits are obviously deviated from the theoretical lines, right? How will you explain it, region? See, ideal state is that the p-value of the expected value completely lies with what we are getting, but it is never so, because the data is not theoretical data, because we are not having there are many missing marker in our data set. It is only when we do not have a missing marker, we have a very good population with a less missing marker, less heterozygosity, and high diversity, then we can expect a very ideal trend. And our aim is actually to find that variant which is away from a common trend, which has a very low p-value. So can you show how LD relate for GCS, please say?
Ma'am, by looking at this QQ plot, we can say that ASI trait is more correlated than others. It is very near to
yeah,
black line.
Yeah, it is there, but if you see it is more correlated, but because it is if you see those SNP which are getting diverged, which are which are having very low p-value and going away from the minus log p-value is much lesser, means higher, so the value is actually much lesser. So that is not so in this yellow, it is just here. But in this pink plot, if you see, it is going very much away. These two.
Okay, ma'am, on this basis, what we can interpret from these results? I didn't
See,
get.
if it is you interpret that these SNPs, because it is actually plotting the SNPs, so it is better if I will explain through Manhattan, because in this it is doesn't show the position of the SNP, it only shows the p-value. If you go to Manhattan, it will explain you that I will make the Manhattan plot. Here I will show in my PPT, it is not getting made. So if you see in the Manhattan plot, it shows you that at which position of the SNP, at which position in your genome of your organism, the SNP, the variant which you are looking, is having very significant p-value, and so it is the most probable SNP which is associated with your trait. That is, it is in LD. We are basically identifying those SNPs only which are in LD, which are in linkage disequilibrium with the traits which we are studying. So I feel you understood this point. Then I will explain you. Someone, ma'am, can you share any paper which give details of R codes for these kinds of analysis? Yes, I will share the paper, and it is very easy to use those things, because once you important is to run the file to get this output stats file. Once you get this, then it is not a tough job. Okay, and Manhattan plot has an advantage that, like, you are plotting it this way, only one way in here in your Tessel, but you can plot in many other ways in R also, because R has many options which you can change. Okay. So now I will be explaining you a little bit about QQ and Manhattan plot in detail. The output of GLM analysis is produced under the result node. The GLM test is evaluated. This association test we evaluated by QQ and Manhattan plot. From the QQ plot, we see that there are several markers that appears to be falsely associated with the trait. Therefore, to control the co-founding effect, there are many markers which are not, there are some which are not actually associated, but falsely associated with the trait. Therefore, to control this co-founding effect, this kingship matrix, which I told you can be used in the MLM analysis, is prepared, and which is a kingship is basically a relatedness matrix based on relatedness, which is another covariate in the linear, because this is all general linear modeling we are doing. So it is an additional covariate. So it supports our data set, and it helps to find those SNPs which are actually associated with the trait and are not falsely associated. So if you want to find the kingship matrix, we can find the kingship here. Someone has asked, what is our aim form? What is the aim form QQ plot? And as I told you so many times, actual p-value should be near or far away from the expected. Those SNP which are far away from the expected are those which are actually having much lower p-value and are in LD. And if it is difficult to understand QQ plot, please understand Manhattan plot, and it is more easier. And if it is used to evaluate the QQ and Manhattan plots are used to evaluate your GLM, that is the model which you have run. How correct is the model? How many false positives are there? So I will be explaining you how to find the kingship matrix. So you will select genotype data, go to kingship maximum allele method. I have been using the centered IBS method, max allele 6. So it has calculated the relatedness between the taxas for the different markers. Once this is created, this is the kingship matrix, and here is your. Now we have to intersect all the files, the genotype, phenotype, and the kingship matrix. Once you intersect that, then you can run the other model. Sorry. Matrix is not selected actually. Yes, okay. So you can do this part, this kingship part, kingship matrix, and then run MLM model also if you want to. Here, because we result are good in GLM, so I'm not running MLM here. You just have to prepare this kingship matrix only. Okay. And if you want to run the PCA of it, I can run the PCA of it.
Hello, ma'am.
Hello.
Hello, ma'am. On what basis we can see that our results are good or not? We have to go for MLM or not?
See, actually, the results which we got in the Manhattan, you have seen that there were SNPs which were which showed that having a significant p-value which were above the expected trend of the all the normal distributed SNPs. So at few chromosome, which I will be explaining you here again, you see that those SNPs are significant and are most likely to be associated with your trait. That's why I went for GLM model and didn't go for kingship further. But still, you can go for kingship if you want to remove the all the major false positive which have come in GLM. Just you have to add this kingship matrix, nothing else you have to do.
And my question is that, like in statistics, mostly people find out p-value is always less than 0.005.
Right.
So this is same threshold find in the genomics analysis finder. So you have any other criteria like you're saying. So you have to go in same like result in how the statistics saying in p-value less than 0.05. There some any other way to analyze in genomic kind of data.
This is the only, this is correct. It is always the p-value is based on 5% or it is based on 1%. Only thing is that because here you see that at the end we get a data set plotted based on p-value. So the cutoff is decided there only, because you see that in your QQ, in your Manhattan plot, that majority of these SNPs at a point are present at a specific, like if you see 4 is the p-value here where majority of the SNPs are present. So if you so those which are above this cutoff at the highest point, they are considered to be the most significant. That is the difference which from the normal statistical test what we are looking at.
Okay, thank you.
Hello, ma'am.
Yes.
Sorry, I would like to know how to choose that 4 is significant or 5 is significant.
4 significant, which 4?
I mean, that you mentioned that on the x-axis there was 4 on Manhattan plot.
Yeah. See the cutoff, if you see the cutoff, I'll let that plot come. I will explain you just now. Yeah, the cutoff you see here, it is cutoff. Majority of the SNPs are in this range only, but those which are going above 4 or 5, 6, this 6, if you see, are very rare. So those are the rare which are show LD compared to the other known other predicted SNPs.
I mean, do we choose ourselves this 4 or this program automatically choose that
No,
4 is?
this cutoff, we put a cutoff line according to the output which we get, but generally it is people take, if you see in papers 4, 4, 5, this is the cutoff which is observed.
Okay. Thank you.
No, here the p-value is 0.05 is cutoff value, but
Yeah.
here showing negative logarithmic p-value. So what is the negative logarithm p-value?
Actually, the values here are given in positive. It is 5 is written. So it says that it is lesser than 1. That's why it is in negative log.
So here showing the highest value, which one is the SNP is more significant. So,
Yes.
but in the p-value is less 0.05.
Lesser than, more negative the value, more significant it is, no?
But here showing highest value more significant in the Manhattan plant.
Yeah, because it has negative log of it. It is not positive log.
Okay.
That's why it is negative log, because whenever you have to represent a value which is less than 1, then you put a negative log so that it comes in the ratio and then you get 0.05 this way. Because p-value is always significant when it is lesser than a specific cutoff like 5%, 0.05, and you said p-value should be less than 0.0, but Manhattan plot shows more than 0.05. Please define clearly.
Yeah, this is the p-value 0.05 more less is significant, but here showing more value, highest value.
Actually, it is positive. If you don't see, I feel you are not able to get what I'm saying. See the value, I will plot again and then show you. if you see here, this value is positive value. If you only see the positive value, 6 is this 6 is minus log 10 to the 6, minus log 10 means it is lesser. The upper value is lesser, not
Okay.
high.
Okay. So it's not going above 0.05. It's going to negative
It is
opposite.
no, see, if it is negative log, then your value is getting lesser and lesser. Na? It is 0.05 or it is lesser than 0.05.
Okay. Okay.
So I feel all of you understood today
Yep.
is lecture.
And madam, one
Ma'am,
more thing.
Yes.
So how to plot linkage disequilibrium for this data?
Okay. Actually, linkage disequilibrium in itself is a separate software. HaploView is a very popular software, and I've explained this so many times. I don't know. Why you can write the proper mail. I will explain you more. So I'm saying that LD value is very popular software. HaploView is used to make a proper LD of your complete data set. So that is a complete in itself a separate lecture. So by here, if you plot LD, it will not give a proper LD what you expect to be made in your figure, which is an ideal HaploView figure. So for that, an additional lecture can be made. I'll ask to the team if possible. Then for some time we can meet and connect, and then I can explain you about LD. Because then I have to prepare those slides. I have not prepared the slide, and it will take time for me to explain you.
hello, ma'am.
Yes.
If we have samples like male, female sample and case control sample, then
Yeah.
in which case male, female should be considered as covariate, or I mean, if we have disease case control, and we would like to check SNPs associated with this disease, should we consider male female as covariate, or should we check disease associated with male female also?
Male are considering male female as covariate is better.
Okay. And in some studies, people get results SNPs like 10,000 SNPs or
Right.
1 million SNPs. Can this value change, like p value change according to number also? I mean, can we
Yeah, see,
select?
that's why I said the cutoff will depend on your data set, that what is the cutoff where your majority SNPs are lying, and those which are above exceptionally above the other SNPs. So
Yeah.
always there is a cutoff, like you have a cutoff line, like you have it is 0.5%, but it varies if you have very high data set, a very high number of SNP. It can be some exceptional p value cutoff you get, and that too when you get many SNP above a specific cutoff, then what you do, that you identify the function of that gene in which that SNP is falling.
Yeah, in this paper that you mentioned, you have shared us, there was like
Yes.
0.05,
Yeah.
0.05 divided by those number of SNPs, and then
Okay.
they had checked this cutoff. So is it a right way?
Yeah, that the paper I exactly have shared are very good papers, and those are, so you can idly follow them. And
Okay,
please
thank you, ma'am.
only take better that if you follow those paper which according to, like, they have done analysis in a case control in human, then follow that paper for your analysis if you are dealing with that set of data. But if you are dealing with some other type of data set, where you are trying to look some other phenotype, then because the organism genome, its ploidy level, and the trait, which type of either is a quantitative or qualitative trait, it all depends, and that all decides what type of parameters should be used. So only use those reference paper. Don't take a human paper reference in plants without considering the data of the plant, because plant genome, a girl was working on plant yesterday, she has a question. So in that cases, many plant are very complex. Their genome is much more complex compared to the human. They are very highly repetitive, have very high polyploidy level, and they also have a very heterozygosity level is also very high. So in that case, you should always take those references, and in that you have to at many steps modify your data. The data set which I have shown you is of a human data set, and for this TESOL, and it is a very it's a maze data set. It's not a human data set, it's a maze data set, and that is a very ideal data set studied by the lab which has prepared this software. So that's why we didn't make many changes.
Okay, ma'am. Just last query. There is this Jiva summary statistics file where
Yeah.
we get chromosome based pair p-value,
Right.
and how to choose that, because sometimes what happens, you have same p-value for multiple chromosome, but you would like to choose the best associated SNPs. How to choose?
Yeah, I told you that if there is a same p-value and for multiple SNPs, this only you want to ask?
Yeah.
Yeah, if there is a same p-value for multiple SNP, then you should surely go on its function and check which the SNP present on which of the genes is more functionally correlated with your trait.
Even the phenotypic variance is very much important.
Yeah, and phenotypic variance. So obviously when you take the phenotype data, you consider the phenotypic variance is good, then only you go with that data set.
No, between two SNPs, based on R square value, she can reduce the.
Okay.
Yeah, that is R square is also a good measure that you can take into account.
Thank you.
Hello, ma'am.
Yeah, hello.
Ma'am, I got confused. Why we consider male and female as a covariate? Because the genomic DNA is almost same.
Why we consider male and female as a covariate? Because her aim was to identify the diseased and non-diseased patient data. Because her study was
patient
based.
also male and female.
Anji?
There will be no mention about a patient male or female.
See, when you add whenever you add an additional information to your genotype data, then you have to, if you how will you give it that both that together? Like you also, like either you are using such a software which will take two types of phenotype together, like the stack which we have used. But if you do not take two phenotypes, because there are two types of observations here for your input individual. One is based on disease, non-diseased, and other is based on male and female. So better to best answer would be to read a such paper. I will share a paper with you in which this is explained very well. Okay, so my class time is over. So nice having a nice session with you all. You all can take my email ID and have when I'll mail the papers, then you can anytime contact me and ask your queries. I would be more than happy. So
okay, ma'am, please share the paper of CML and JLM.
Yeah.
Actually, maybe having time late.
Yeah,
maybe
decode.
like
Can I connect to decode?
Yes, ma'am.
Yeah. So I can conclude. Stop the class here.
Yes, ma'am.
Okay. Thank you so much.
So thanks, ma'am, and thanks for your time and your wonderful sessions. We believe our participants gained with your experiences, and we are thankful that you deliver on our platform. It is a matter of honor for us. So
yeah, sure.
And we will again invite you in our future events.
Yeah. Someone is asking to arrange a session on LD, and this it can be arranged separately whenever next
yes,
time.
ma'am. So what in the feedback form, we will ask about what they want to learn in genome-wide association studies. Then after recommendation of our participant, we can design a module. And so first we will ask for their recommendation, what actually they want to learn, and then we will again plan a comprehensive bioinformatics workshop on genome-wide association studies. So this is the first time, and we have only planned 5 lectures for genome-wide association studies, but in future we will try to make it more comprehensive so that more and more people became or get to know about these genome-wide association studies.
Yes, please make it, because in these 5 lectures we just get an idea about association method.
So definitely we are going to
These
have
are,
more
because almost 15 days we have spent on their modeling and other. Please complete.
So thanks, ma'am, and thanks for your time. Now I will seek permission for ending today's session, ma'am.
Yes, yes. Thank you so much. Thanks. I had a great experience. I can be always available for you. So thank you very much.
And there is a minor announcement. Tomorrow is session, rescheduled session on Python project. So timing will be again 7 p.m. Indian Standard Time. So if you want to attend the project session on Python classes, session will be tomorrow at 7 p.m. Indian Standard Time. So thanks.
And sir, where that Dr. Priyanka is, where she was working, because I don't know about her, so where I can get a guide from her.
Sir, I'm not getting you. Can you speak loudly?
I'm asking the Dr. Priyanka is teaching last 3 classes. So which institute she was working aware, because we are not I'm not able to get where she is, like I can contact for any future query and all those things.
So she's working as a genomics officer in a well-reputed institute. Kindly email me. I will send her details. Okay.
Sure. Okay.
Thank you.